Assalamu alaikum dear students. Today we are talking about KMU-CAT 2025 test preparation part 1F. Dear students, if you need MCQs for KMU-CAT test preparation, let me know in the comment section. अगर आप KMU-CAT टेस्ट के लिए तैयारी कर रहे हैं और आपको MCQs चाहिए तो आप कमेंट सेक्शन में बता सकते हैं. Dear viewers, here you can see rescheduling KMU-CAT centralized admission test 2025-2026. Dear students, अगर अभी तक आपने टेस्ट के लिए रजिस्ट्रेशन नहीं किए तो जल्दी से रजिस्ट्रेशन कर लें और अपना सीट कंफर्म कर लें. अगर आप KMU-CAT टेस्ट के लिए तैयारी कर रहे हैं तो ये वीडियो आपके लिए है. इस वीडियो को आखिर तक ज़रूर देखें. What differentiates each amino acid group is option A, the carboxyl group, B, hydrogen bond, C, charge, and option D, the R group. So the correct option is D, R group. Explanation is side chains means R groups give each amino acid unique properties. So this is very important MCQs. Deamination is dash of an amino acid group. Option A, removal, B, addition, C, supplementation, and option D, none of these. So the correct option is A, removal. Deamination means removal of amino group. So deamination remove amino group from amino acids. Subscribe now and press the bell icon. Never miss an update from a technologist. So this is very important MCQs. Proteins produce polypeptide from proteins by option A, oxidizing, B, reducing, C, hydrolysis, and option D, none of these. So the correct option is C, hydrolysis. Hydrolysis breaks peptide bonds to form smaller peptides. The disulfide bond is not broken under the usual condition of option A, filtration, B, reduction, C, oxidation, and option D, denaturation. So the correct option is D, denaturation. Disulfide bonds require reducing agents to break down the bonds, not just denaturation. Each hydrogen bond is quite option A, weak, B, strong, C, both A and B, and option D, none of these. So the correct option is A, weak. In an individual, hydrogen bonds are weak but collectively stabilize structures. So dear students, this is very important MCQs. In many proteins, the hydrogen bonding produces a regular coiled arrangement called option A, alpha helix, B, beta helix, C, both A and B, and option D, none of these. So the correct option is A, alpha helix. The alpha helix is a common secondary structure stabilized by hydrogen bonds. Many globular proteins are stable in solution, although they lack option A, hydrogen bonds, B, oxidation, C, nonpolar bonds, and option D, disulfide bonds. So the correct option is D, disulfide bonds. Explanation is unlike fibrous proteins, many globular proteins don't need disulfide bonds. Each turn of the alpha helix contains how many amino acids? Option A 2.8, B 3.2, C 3.4 and option D 3.6. So the correct option is D 3.6. The alpha helix has 3.6 residues per turn. So this is very important MCQs. Which of the following protein structures looks like a telephone card? Option A primary structure, B secondary structure, C tertiary structure and option D quaternary structure. So the correct option is B secondary structure. The alpha helix resembles a coiled like telephone cord. Dear students, this is very important MCQs. The hydrogen bonds in the secondary and tertiary structure of proteins are directly attached by option A salts, B alkalis, C detergents and option D all of these. So the correct option is D all of these. So salts, alkalis and detergents can all disturb hydrogen bonds. So this is very important MCQs. The alpha helix is stabilized by option A hydrogen bonds, B disulfide bonds, C salt bonds and option D non-polar bonds. So the correct option is A hydrogen bond. Hydrogen bonds between backbone groups stabilize the alpha helix. So insulin controls blood sugar level and storage of glycogen and is produced in option A pancreas, B pituitary gland, C adrenal gland and option D parathyroid glands. So the correct option is A pancreas. Insulin is produced by pancreatic beta cells. Proteins contain option A only L amino acids, B only D amino acids, C DL amino acids and option D both A and B. So the correct option is A only L amino acids. Explanation is with rare exceptions proteins contain only L amino acids. If you like this video, please सब्सक्राइब माई यूट्यूब चैनल। अगर आपको ये वीडियो पसंद है बराए मेहरबानी मेरे यूट्यूब चैनल को सब्सक्राइब, लाइक और शेयर ज़रूर कर लें। द इंडिजेनिक एंटीबॉडी फंक्शंस ऑफ प्रोटीन्स बाय डिनेचरेशन आर फ्रीक्वेंटली। ऑप्शन ए नॉट चेंज, बी चेंज्ड, C both A and B and option D none of these. So the correct option is B changed. Denaturation typically destroy antibody binding sites. Contractile proteins used in muscle contractions are called option A collagen, B immunoglobulin, C elastin and option D actin and myosin. So the correct option is D actin and myosin. So explanation is these two proteins interact to enable muscle contraction. Thank you for watching my videos.